Okay, so I hope you use your time wisely. Now remember, I'm gonna ask the cover teacher to go over to just check in terms of how you guys are doing, and he or she will be checking in your work. Now, so in this case, let's look at this. I'm just gonna use the answer key one to just see if I can zoom in a little bit more. Oops, perfect. Now again, so I'm just doing the wrap up. Now remember, this lesson has three important parts. One of them is talk about the nucleus, the genetic material inside the nucleus. So let's talk about that part first. So most of the time, the genetic materials are here, and in this case, yeah, just go over there. So your genetic materials in the nucleus is actually like this. Is kind of it's all spread out now. So in the spread out form, now keep in mind, kind of skim through a little bit ahead of time. So most of the time in the cell cycle, most of the time the cell is doing what it's supposed to do, right? During most of the time, it's an interphase. So G1, S, and G2. So during that time, the cell is just doing its thing, carrying out its own function, um, making copies of itself, etc., etc. So you do need these genetic material, your DNA, to be unwind, to be spread out. Because if it's not spread out, you can't read them. Okay? So it's all unwind, spread out like this. So when it's in the spread out form, you call them chromatin. Okay? The chromatin. Now, when the cell is gonna be, it's getting ready to be duplicated and to be split, they actually would get these chromatin and wind them up very tightly. So in this case, it's very much like when you're using your charger cord in the lesson. You unwind your charger cord, you let it spread out, you plug in your laptop, and then, oh, there's only 5 minutes before class, so I better pack up now. So I'll take my charger cord, I'll wound it up so it's easy to put my bag. So same kind of idea. Your chromatin, which is spread out DNA, it's gonna wind up into something called chromosome. Okay, so you see this item here, chromosome. So one chromosome is really tightly wound up DNA, so it's easy to split, easy to be moving around. And one chromosome, as you see here, is made up of two copies of what you called chromatid or chromatid. So this chromatid are identical information, and they're joined at this point here called centromere. Okay, centromere. So one chromosome have two sister chromatids. They're called sister because that's identical, and then you have a centromere here. So when you draw it, it's almost like a little butterfly, so to speak. One butterfly, one part, one part, one part, one part like this, and join in the center. Okay. So now each human have 46 chromosomes in your cell, regardless of whatever cell it is, except red blood cell. Half of them is from mom, half of them is from dad. Okay. So here are the terms. Double check your answers on this one. Okay. Now we talk about one of the important part was about what's going on in the nucleus and the terms. Now the next part here, the cell cycle. Now I just talked a little bit about that. Most of the time, your cell is in what you call interphase. So it's G1, growth 1, S, synthesis, G2, and then synthesis, and then G2, which is growth phase 2. Now, growth phase 1 basically is the cell doing whatever it's doing. In the S phase, synthesis phase, your DNA is being replicated. So new strand of DNA is being synthesized, G2, it's at this point, the cell is actually getting ready to go through mitosis. So they will be making more organelles. It will be making more organelles. You'll be double checking all the parts of the DNA, etc., etc. So that's what's going on during the interphase. So you make sure you remember G1, S, and G2. Now, some cells, like in the video, talk about going to a G0 phase, which the cell cycle stops. Okay, we don't look at it that much. We mainly focus on this part. Now, the last but not least is going through the mitosis phase. Okay, so now make sure you check your answer for this. Now, mitosis. So I'm going to enlarge this and draw. So hopefully you can see this a little bit better. See if we can do this. Sorry, guys. So now you have prophase, metaphase, anaphase, and telophase. So during that time, you should distinctly see chromosomes. So all those chromatins, it's going to condense and wound up into chromosomes. They're going to start to gather around in the middle. The nuclear envelope, which was supposed to make up the nucleus, is going to break down. And you have these spindle fibers and made of tiny microtubules. So all these lines are going to join to where the centromeres are, centrosomes or centromeres are in the chromosomes. Okay, so think back to this here. They're all going to join at the centromeres. Now, metaphase is the part where all these chromosomes are going to line up like this in what they call equator, so middle part, in the middle of the cell. And these are at top. These are what you call the centrioles. Okay, it's not shown in the diagram. Doesn't matter. Doesn't matter at this level right now. Next year in grade 11 and 12, you need to know more. Now, at this point, so metaphase is all these chromosomes line up in the middle. That's a main key point. Now, at the anaphase, now remember when we talk about the chromosomes, I kind of said, it's like a little butterfly kind of joined in the middle like this, right? So what happened in the anaphase, quite graphically, so to speak, is these butterflies get split up. So these microtubules actually contracts and then pulls the chromosomes apart. So you now have two sister chromatids. Now these sister chromatids are going to go to their opposite end of the cell, so arrives at each pole. So you have an identical set of chromosomes that arrive. Now should be, what you should say is identical set of sister chromatid arrives at each pole. And now telophase, you're going to start to see the nuclear membrane. So the new nucleus is starting to be forming. Each side of the cell, you will have the sister chromatids, respectively. They're going to start to unwind. They're going to form new nucleus. And it's basically getting ready to go into what you call the cytokinesis. So which is you splitting up one cell, splitting into two genetically identical daughter cells. Okay, so we just talk about here. So make sure you understand this and make sure you check your answers. Now the cover teacher should be able to share this with you or share the link with you. So make sure you check your answers. I also want you to prepare, because next lesson you'll have an entrance ticket. And to talk about magnifications, Next lessons, we're going to be doing microscope lab. But also another thing is that I'm going to add a Google form. I want you to basically on the Google form tell me what do you understand about this worksheet about cell cycle, about nucleus, about cell cycle, and about mitosis. I want you to tell me what you understand, what you learned from it, and of course, if there's any questions. Okay, what do you feel that you could use some more explanation? Because you're only watching a video, you're not hearing me explain anything. So I want to allow you to ask questions like that, and then we can go over it when I come back on Thursday. Okay, so um, so the cover teacher will check things in on you and get ready for the entrance ticket next lesson and get ready to do lab. Okay, have a good day. Bye.